Good day, class. I hope everyone is safe in this time. Today, we will be discussing microbiological safety and control of microorganisms through sterilization and disinfection. Now, the antibiotics part will be discussed separately. Now, the basis of microbiology safety po relies upon the universal precaution po established by the Centers for Disease Control, which states that you need to treat blood and all body fluids po except for sweat as potentially infectious. Now, class is in fact true, no, because these samples po may have unknown microorganisms that are likely to be pathogenic to us, the laboratory scientists. Now, and our role po, no, is to isolate and identify them with strict compliance po to proper safety protocols and control measures po, no, to be able to maintain accuracy in reporting. Now, one of the important types of precaution that we need to adhere, no, based on the microbiological safety is the transmissions-based precautions. Now, this includes contact precaution, droplet precautions, and airborne precautions. So when we say contact precautions, these are precautions that used to stop the spread of infectious agents po na pwede pong matransmit, no, through direct or indirect contact with the patient or with the patient's environment. Now, examples of organisms that you would be handling, no, that needs contact precaution is the multi-drug resistant organisms such as your vancomycin resistant enterococci, your methicillin resistant staphylococcus aureus, and your Clostridium difficile. Now, another transmission-based precaution would be your droplet precaution. Now, this is intended naman po to stop the spread of infectious agents that can be transmitted po by close respiratory contact or by exposure of mucous membranes po to respiratory secretions. Now, examples of bacteria naman that needs droplet precautions are the following. These are your Neisseria meningitidis, which is the causative agent of meningococcemia, your Bordetella pertussis, no, which is a causative agent of whooping cough or pertussis, and your influenza virus, which is an example of a virus. Okay. Now, the other transmission-based precaution naman would be your airborne precaution. So when we say airborne precautions, these are precautions that are used po naman um, to infectious agents po, no, such as your Mycobacterium tuberculosis, which is the causative agent of um, your tuberculosis or your MTB. Um, Another example is your varicella virus and your rubiola virus, which these microorganisms po can remain airborne, okay, and infectious over long distance. So, um, do not be confused po when we say droplet and airborne precautions. So both of them po can infect or can infect the respiratory tract, no? Ang pinagkaiba lang po nila is that droplet precautions po, these are organisms that can lodge themselves on small amounts of waters, which is known as your droplets, no? And usually po they are transmitted on a limited distance, for example, a meter or two. So when we say airborne precautions naman, these are microorganisms that can desiccate and can be spread alone, no, using air, no, or transmitted through air po and can travel long distance. That's why airborne precautions or microorganisms that tends to be transmitted airborne po, mas mataas po yung kanilang safety precautions when it comes to droplet precautions naman. Okay. So how do we know the different hazards po entailed with processing microbiological samples and potentially be exposed, no, to this infectious microorganisms. So dito po papasok yung tinatawag na biological risk assessment. Now, the biological risk assessment po is a process, no, used to recognize the hazardous characteristics of infectious agents po that may be encountered in the clinical microbiology laboratory. So it includes risk assessment process, no, um, uh, that could result po, no, in an infectious exposure. So mga laboratory practices po ito that likelihood that a laboratory acquired infection will occur and the consequences of that infection po. So through this process po, appropriate safety practices po can be identified to protect laboratorians. Okay? So these are the different classification of infective microorganisms by risk group. Now, ito po yung risk group 1, risk group 2, risk group 3, and risk group 4. So dito po na nagbe-base po sa sa susunod po na discussion would be the biosafety levels naman. So, so tandaan po ha, pagka risk groups po, so usually these are classifications, no? Of based on kung ano po yung pathogens na hinahawakan ninyo. So is it risky to handle because it can infect you or the community and so on. So when we say risk group 1, so these are microorganisms po that is unlikely to cause human or animal disease. It has no or low individual and community risk. So when we say risk group 2 po, it is a moderate individual risk naman and then low community risk. So these pathogens po can cause human or animal disease po, pero unlikely to cause serious hazard to laboratory workers, to the community, the livestock po, and the environment. So usually po, laboratory exposures may cause serious infection, yes, but um, uh, existing naman po yung effective treatment and preventive measures po. Okay, and then the risk of spread of infection is limited. So that is risk group 2. Now, when we say risk group 3 naman po, these are pathogens po that have high individual risk but low community risk. Ibig sabihin po, it can cause severe symptoms or signs and symptoms, but then again, it has a low transmission to the community. So it is a pathogen that usually cause serious human or animal disease, pero they are not ordinarily po spread from one infected individual to another. And meron pa rin pong existing na effective treatment and preventive measures for this organism. So, and then lastly, when we say risk group 4 na po, so these are organisms naman po or pathogens that have both high individual and community risk. Ibig sabihin po, usually po, they can cause serious human or animal disease, and hindi po existent on that time po yung treatment po or effective preventive measures po to acquire this organism. Okay, and then traditionally then this is easily transmitted from one individual to another. So these are your organisms under risk group 4. Now, let's talk about po biosafety and biosafety level naman. Now, when we see biosafety level, in the most easiest way to explain it is that biosafety level po is the level of containment naman. When we say level of containment, these are levels po on how you effectively contain the infectious materials that you are going to handle, and based on the different determinants of the level of containment. So when we say biosafety again, the meaning of that is the application of safety precautions po that reduce a laboratory's risk of exposure to that potentially infectious microbe. So, and limit contamination po of the work environment and ultimately the community. So what are the primary risk po that determines the levels of containment? Okay, so this includes the infectivity of the organism that you are going to handle, the severity of the disease that it will cause, the transmissibility, is it easily transmitted, no, or not? Okay, and the nature of the work conducted also, you do you need longer exposure of exposure longer of exposure po to that organism, or low lang po, or uh, does your work activities include high aerosolization of these microbes or not? And syempre din po, the origin of the microbe. Is the microbe po solely infectious to animals, or is it solely infectious to humans? And lastly po is the route of exposure. So, also it need to be considered. So one example of this is that, is it the organism po easily transmitted to respiratory tract, which includes the vital organ, the lungs, or is it through direct contact, no, which pose a lower risk? So that would be the primary risk that determines the levels of containment. Okay, and um, as you can see here po, there are 4 levels of biosafety. Then just like your bio risk levels. Okay, so as you increase the biosafety level, you also need to increase the containment po for that high risk microbes. So, and then um, please take note that each biosafety level po has its own specific containment controls that requires the following, no? So different laboratory practices, enhanced safety equipments, and also the facility construction. So yun yung po yung tatlong points, no, na nagbabago, no, in every biosafety level. The laboratory practices, the safety equipment, and also the facility construction. On the next slide naman po, nakatabulate na po yung different biosafety levels natin and the containment that you need to know. Now, in this table po shows you the summary of the different biosafety level po and the risk groups entailed to these biosafety levels and the representative organisms po under different biosafety levels. If you want to know the full list of organisms per biosafety levels, you can see it under the website of the Centers for Disease Control. Okay. Now, let me explain lang po shortly the different biosafety levels. Now, the biosafety level 1 includes the organisms under risk group 1, okay, which includes Bacillus subtilis or Naegleria gruberi, no, and other organisms po that have low individual and low community risk. Now, in terms of containment equipment like your biosafety cabinet requirement, now for BSL1 po, it doesn't require any biosafety cabinet. Okay? So usually po, primary barriers also are not required in biosafety level 1 laboratories, no? And then usually secondary barriers lang po includes the general good laboratory practice and hand washing. Now, additional practices po under biosafety level 1 one includes limited laboratory access, the proper decontamination procedures, and then in addition, surfaces that are easily cleaned and not porous po. Usually, ganun lang yung laboratory practices in biosafety level 1. And the work po in the biosafety level 1 can be done, no, in a bench top. Okay, ibig sabihin po on the table lang po. You can manipulate the organisms under that laboratory level or biosafety level po on benchtop. Now, in biosafety level 2 naman, these are agents naman po that are considered as risk group 2. Okay, so kung makikita nyo, parehas lang naman yung usually yung risk group 2, the risk group number is also under the same number or same level in biosafety. Now, in biosafety level 2, these are organisms that have moderate individual and low community risk. And this includes broad spectrum indigenous moderate risk agents. And then some of the representative agents include your viruses HIV, your Hep B virus, your organism like Salmonella, okay, and Toxoplasma. So this includes also um, your Staphylococcus aureus. This is under biosafety level. In biosafety level 1 po pala, one example of a bacteria that you are going to hear, no? Most frequently is your Escherichia coli. Your Escherichia coli is under biosafety level 1. Okay? Now, in biosafety level 2 po, required na po yung pagkakaroon, no, ng biosafety cabinet level or class 2 or other primary barrier when procedures may produce aerosols or splashes. Kasi mostly on biosafety level 2 laboratory, usually yung mga activities na dyan or yung test that you are going to do can generate, no, splashes and aerosols. So for you to be protected, no, dapat you are working na under a biosafety cabinet. Okay? So we will discuss po later kung ano po ba yung mga biosafety cabinets and the different classes. Now, in the biosafety level 2 din po, so primary BSC2 as indicated or other equipments that prevents the aerosolization is required. And also, it requires the use of personal protective equipment as appropriate. Now, in biosafety level 2, second barriers po or practices, the practices po is same with your biosafety level 1. So, kailangan mo ng good laboratory practice and hand washing. And added to this would be there should be evident, no, of proper waste decontamination and disposal of lalo na po yung mga disposable equipment na ginagamit nyo and apparatus. And then there should be evidence of or presence of safety equipment, like for example, eyewear station, and then also it needs pest management program. So the additional practices in BSL2 naman is same as the biosafety level 1, but also it needs to include na po as biohazard symbols, no, posted on the entrance. So and also it needs immunization requirement for the employees and medical surveillance for exposure po of the personnel. And also it needs training naman, specific personal training po requirement for the laboratory workers. And lastly, the biosafety level 2 laboratories needs to have a biosafety manual po including incident and exposure procedures, no? So established by the laboratory manager. Now in biosafety level 3 naman po, these are organisms naman po under risk group 3. No, these are organisms that have very high individual risk but low community risk. So it includes po indigenous or exotic agents with potential respiratory transmission. Okay? Representative agents po include your Mycobacterium tuberculosis needs to be manipulated in a biosafety level 3 laboratory. Okay? And your fungi also, lalo na po yung ating mold stages, no? It needs to be manipulated under biosafety level 3. Okay? And your biosafety level 3 laboratories needs to have biosafety cabinets 2 or 3. Okay? And then the primary barriers for BSL 3 is the same as in biosafety level 2, but additionally it should have, no, ano na po, gas tight aerosol generation chambers na po yan. So, kailangan yung air po sa loob, hindi po siya dapat lalabas in any way possible. Okay? And then, PPE is solid front or wrap around, and then scrub suits or coveralls that are not removed from the laboratory. So, mas pinataas lang po yung personal protective equipments ninyo. And then, for the secondary barrier po, same as with BSL2, and then additionally, it needs to have a ventilation requirement. No? Usually po, yung ventilation po the airflow should be one way pagka BSL3 na. And then, usually po, your biosafety level 3 na po includes what we known as your anterooms, no? Yung mga, ano po yan, may area po that is separated from the main laboratory, mga self-closing doors, okay? And all windows must be sealed in biosafety level 3. And then access is limited using an anteroom nga po to prevent exchange with outside areas of the laboratory. And then additionally, your biosafety level 3 needs to what? You need to routinely check the personnel's serum samples, no, for seroconversion comparisons to monitor exposure. And then lastly, the highest containment po would be your biosafety level 4. No, ito naman po yung mga agents that are under risk group 4 and then highly likely po to cause serious or lethal disease, no? And it's not generally known to be preventable or treatable. So examples of these are your dangerous and exotic agents. Now, examples of these are the hemorrhagic viruses, your Marburg viruses, your Ebola, no, and then your Congo Crimean hemorrhagic fever. And then, tandaan nyo lang po, in biosafety level 4, biosafety class 3 is a must. So, dapat a 3 po na yung ginagamit for biosafety level 4 laboratory. And kailangan po your personnel po, no, is under a full body self-supplied or air-supplied positive pressure suits na tinatawag. Yun po yung personnel po natin ay nakasuot ng um, pang-astronaut na personal protective equipment na may sariling supply po ng hangin. Okay? And then for the secondary barrier of BSL4 po, it needs to have a special zone ventilation systems, usually negative pressure air is supplied, and then all items po are processed through a fumigation chamber or airlock system. And then personal decontamination procedures included such as your showering or chemical treatment should be done before you go out on that facility, the BSL 4 facility. And the in and out of the personnel or items entering and leaving the laboratory should be consistently logged. Okay? And then generally includes complex waste management procedures po ang ating BSL 4. Now, so this slide po provide you a better view, no, on different biosafety levels po. So how do you recognize easily different biosafety levels? So tignan nyo lang po kung paano po nag-work yung isang scientist and ano yung mga personal protective equipments mo na ginagamit nila. So as you can see here in biosafety level 1 po, you can work on bench top and then personal protective equipment is minimal. You don't need respiratory protections and also um, splash protection equipment. Now for BSL2 naman, as you can see here, PPE is fully worn, including lab coats and gloves. Eye protection and face shields can be also worn as needed. And then all procedures po that can cause infection from aerosols or splashes are performed under a biosafety cabinet. Yung naka-include po dito sa letter B in BSL2. Okay? And then it should be noted also in biosafety level 2 facility, an autoclave or an alternative method po of decontamination is available uh, for proper disposal. Now, lastly, in BSL2, the laboratory should have self-closing doors. Now, in BSL3, as you can see here na, mas pinataas na po yung personal protective equipment, respirators might be required, no? And appropriate biosafety cabinet should be evident. And then entrance to the laboratory is through two sets of self-closing and locking doors po, okay? To enhance naman the security, no, biosafety, biosecurity, no, of the facility. And lastly, the highest containment is the biosafety level 4, which pinaka-appropriate po na ginagamit na class ng biosafety cabinet would be biosafety cabinet class 3. No, and then the personnel should wear a full body air supplied positive pressure suits. Okay? Tinatawag rin po itong hazmat suit. Okay? Your hazmat suits. Okay. So let's now discuss now the different biosafety cabinets. So kanina pa po natin nasasabi, ano po ba yung biosafety cabinets? So generally, class, the biosafety cabinets po are hoods, no? They are like chemical hoods or fume hoods. Usually po, they are specialized po when it comes to providing protection, no, against biological hazards. Okay? This is a type of containment barrier po that protects the worker from the aerosolized transmission of organisms. Okay? And there are different classes of biosafety cabinet, namely biosafety class 1, class 2, and class 3. Okay? So the main or the basis of protection po of biosafety cabinets po is that it provides po um, a controlled airflow po within the safety cabinet po that shields you from the aerosolization of your potentially infectious po na organism. So by providing a curtain of air, which is usually controlled via a system of a blower, no, inside the biosafety cabinet. Okay? Now, in terms of classes, so the class 1 biosafety cabinet po uses an exhaust fan, no, to move air inward to the open front. So tandaan niyo po, so please take note that biosafety cabinet class 1 and class 2 are classes of biosafety cabinets po that have an open front type of cabinet. Pag sinabing open front type, window type po yung kanilang opening sa harap. Okay, naaangat po yung kanyang glass door and then allowing you to have an open space or a designated space po to work with. Okay? So, yan yan. Now, the air in the Class 1 BSC po is circulated po within the safety hood. Now, passing a type of specialized filter known as your HEPA filter or your high efficiency particulate air filter. Now, tandaan nyo po, no, your biosafety or biological safety cabinets, the air po that must be flowing inside po should be filtered, okay? Pero depende pa rin po yan sa class ng biosafety cabinet. And the most standardized filter that is used on our biosafety cabinets po is the HEPA filter, your high efficiency particulate air filter. Okay? Now, the air will go in the HEPA filter before reaching the environment outside the hood. Ang kinaibahan lang po ng class 1 to other classes of biosafety cabinet is that the as you can see here, the air po that is supplied inside the biosafety cabinet is not filtered by the HEPA filter. Isa lang po yung filtered po niya at ang pinefilter lang po niya is the air going outside. No? Kasi nga po, the air should be decontaminated by filtering system po using a HEPA filter. Okay? So usually class 1 BSC is now obsolete. Okay? Ibig sabihin hindi na po ito ginagamit kasi it can only provide personal protection and environment protection, but it cannot provide a protection to the samples that you are going to work. Ang tandaan nyo po, ang pinaka ina-achieve din natin po is dapat hindi po na co-contaminate po yung ating samples. Dapat yung representative um, bacteria or microorganism lang po natin, yun dapat tinetest natin. And contamination from other microorganisms that are not necessarily to be reported, dapat po iwasan natin yun. At hindi yun naiiwasan ng ating biosafety class 1. Okay? Okay. Now, for biosafety cabinet class 2 po. So, this is the most common in microbiology laboratories nowadays. Now, specifically po, biosafety class 2 A2 is the most commonly acquired and used in the laboratories nowadays. Now, in this type of biosafety or class of biosafety po, both um, the supplied air and the exhausted air po is filtered. Pero iba lang po yung percent po na nire-recirculate at nilalabas. Okay? So air is pulled inward and downward by a blower and pass up through the airflow plenum po, where yung air po, bago po lumabas po, is filtered. Okay? And in class 2 BSC, kaya po siya yung pinakaginagamit na ngayon is that there is a percentage of the remaining air po that is HEPA filtered po na nire-recirculate at nilalabas ng biosafety cabinet. Ibig sabihin lang po, the recirculation inside the biosafety class 2 po is necessary po for you to increase the protection by the aerosolization because it can supply a V-shape curtain of air po in the open front plenum. Okay? So, ibig sabihin po, hindi ka masyado mababahala kung yung work mo under that cabinet po is nag-generate ng too much aerosol. Kasi yung aerosol, hindi po siya lalabas on the open front. Kasi may nakaharang po na curtain of air provided um, by the system of your biosafety class 2. Okay? So, yung maduming air po, magpupunta lang po siya doon sa taas ng blower wherein another filter po should decontaminate it and then bago po lumabas doon sa pinakataas ng biosafety cabinet. So, usually po, biosafety class 2 po has two sets of HEPA filter. So, yung isang HEPA filter po will filter the supplied air inside the cabinet and another filter po should filter naman yung air that will go outside the biosafety cabinet, providing complete protection. It provides personal protection, environment protection, and also sample protection, okay, or product protection. Now, lastly, this is the highest class of biosafety po na cabinet. Okay, this is your biosafety cabinet class 3. And the difference between class 3 and the previously mentioned two classes of biosafety cabinet is that your class 3 biosafety cabinet is not an open front type of cabinet. So as you can see here po, the um, the cabinet po, the Class 3 biosafety cabinet is fully enclosed, okay? So as you can see here nga po, no, may sarili po siyang gloves, no, na naka-provide, no? And then, it is a self-contained ventilated system, okay? This is for handling po highly infectious microorganisms na hindi kailangan po dapat nalalabas in any way possible doon sa cabinet mo. Okay? So the closed front contains attached gloves, no, for manipulation of the work surface inside the cabinet. So kaya nga po tinatawag ito na gloved boxes. And provided here also is the pass box. So bago pumasok yung mga gamit, dapat papasok muna siya sa pass box para um, wala pong contamination from outside environment and the inside environment. Kaya para po ma-maintain yung sterility na tinatawag. Okay? And also, the provided air here, bago pumasok, it is completely filtered by a HEPA filter, yung supplied air. So as you can see here on the right side of the cabinet. And then, the supplied air, lahat po ng air po, hindi po dapat nire-recirculate. Hindi pwedeng i-recirculate po yan. So, kailangan one-way flow lang po yung air. So, one flow from the HEPA filter, and then yung lalabas po na air, it is pass through in another HEPA filter before going outside the environment. Okay? So, ganyan po kataas yung level of containment ng Class 3 BSC. That's why Biosafety Class 3 po is used under Biosafety Level 3 and 4 facilities. Now, additionally, class, your Class 2 Biosafety Cabinet has different types then. Okay? So, what are the different types of your Class 2 BSC? So, this includes your Class 2A1, Class 2A2, Class 2B1, and Class 2B2. So also, no, you can include the Class 2C1, but different manufacturers po usually exclude na yung Class 2C1 kasi halos kaparehas lang po siya ng Type A cabinets. Okay, yung ating Type C. So, and also type B as well. Okay? So, kaya po yung four common types lang po ng class 2 would be class 2A1, class 2A2, 2B1, and 2B2. So, let us see po ano po ba yung mga difference ng mga classes of biosafety cabinet. Tandaan niyo po ulit ha, please take note. Routinely used po na class ng biosafety cabinet would be biosafety cabinet 2A2. Okay? Ang 2A2 previously po na NSF classification niya would be class 2 type A or type B3. So, yan. Okay. So, let us know po what are the different parameters po na ginagamit natin to differentiate the different classes po of your biosafety cabinet. So, this includes the type of protection that they provide, the risk groups that are that can be manipulated or worked with in that class of biosafety cabinet. Um, the third parameter is that if the biosafety cabinet po ba can be used for volatile radionuclide or chemical protection, it can provide chemical protection ba? Pang-apat po is that ano po ba yung supplied air po niya tsaka exhaust air po niya? Is it filtered or not HEPA filtered? Okay? And next naman po is the phase velocity. So when we say phase velocity class, usually it is applicable lang po for biosafety cabinets po that have open front window. Okay, an open front type biosafety cabinet. That includes class 1 and class 2. So when we say phase velocity po, kung gaano lang po kabilis or gaano kadami yung sinusupply na air po niya using the blower. Okay? And then, next naman po is the percentage of the air that is exhausted and also recirculated in the biosafety cabinet. And lastly is the exhaust system. Pag sinabing exhaust system, paano po lumalabas yung hangin doon sa biosafety cabinet? Is it hard ducted? When we say hard ducted, the air is completely thrown out of the building, no, and then in the environment. And then when we say room exhaust system naman or thimble connection, yung air po can be thrown in the room itself, okay, without using a without using a duct. Okay? So as you can see here lang po, in terms of type of protection class, the lowest type of class of biosafety cabinet, your class 1, no, it can only protect the personal and environment. While your class 2 and class 3 po can provide complete protection. It can provide personnel, product, and environment protection. Now, in terms of risk groups po that you can handle in the different classes of biosafety cabinet. So, for your class 1 po, you can handle risk groups 1, 2, 3, and 4. If aabot po siya ng 3 and 4 if you are um, in the proper PPE. Usually po, if the personnel po is under a suit laboratory. Okay? But if you are not under a proper PPE, you can only handle risk groups 1 and 2 in class 1. Na in class 2 naman po, you can handle po risk groups 1, 2, 3, and 4. So it can go up to 4 provided na you are under a positive pressure suit. Okay? So if you are not under a positive pressure suit class, you can only work until risk group 3 in class 2 biosafety cabinet. Now in in class 3 biosafety naman po na cabinet, you can work po risk groups 1, 2, 3, and 4. So kompleto naman siya. Now in terms of chemical protection and protection from toxic substances, including toxic vapors po, class 2A1 and 2A2 po cannot provide no proper protection in chemical protection and toxic vapors because tingnan niyo po yung kanyang exhaust system po. So, class 2A1 and class 2A2 po, ang kanyang exhaust system po is room. Okay? Through room. Ibig sabihin po, toxic substances kasi cannot be exhausted in the room itself. It can harm. Okay, it is hazardous po to the worker. That's why only class 2A1 and A2 po cannot provide chemical protection and protection against toxic volatile substances. While the class 1, B1, B2, and class 3 po can provide protection against toxic substances. Okay, because their exhaust system is hard ducted. Okay? Now, in terms of the supplied and exhaust air, is it HEPA filtered po ba or not? The only biosafety class po that do not supply HEPA filtered air po is class 1. The rest po, both supplied air and exhaust air po is HEPA filtered. Now, in terms of phase velocity, the lowest phase velocity po is class 1. Okay? Kasi po ano lang po, mahina lang yung blower niya. While the class 2 A1 and A1 po has 0.38 meters per second na phase velocity and the other class 2 subtypes, your A2, B1, and B2 po, has the same phase velocity. Now, as you can see here po, class 3 po do not have phase velocity because then again, your class 3 po is not an open front cabinet. It is a closed type of cabinet. Okay? And in terms of the percentage po na exhausted naman and recirculated, as you can see here, only class 1, class 2 B1, B2, and 3 po, yung may class 1, class B2, and 3 po, yung wala pong nare-recirculate na air. Okay, tandaan nyo, class 1, class 2 B2, and class 3 po are the only cabinets po that do not recirculate air. Okay, yan yun. And then for the others, your class 2 A1, A2, and B1 po, meron po silang percent na nare-recirculate. Okay, for class 2 A1, A2, pareho po yung percentage exhausted nila. It exhausts 30% of the air and it recirculates 70% of the air. While your B1 naman po, more than 50% of the air is exhausted and less than 50% naman po is recirculated. Okay? And lastly, so class 1, class 2B1, B2, and class 3 po are the cabinets po that are hard ducted. Ibig sabihin, sa taas po ng cabinet po nila, it has a large hose or exhaust or duct that exhaust air completely outside the building, okay, or outside the facility. So that are the difference between the classes of biosafety cabinets. Okay. So in this slide naman po shows you the simple engineering or the different parts no of your class 2 cabinet. Okay. So please take note class high, your biosafety cabinets have two basic parts for its protection capability. So this is your blower no and also the filter. Okay. Doon po nakadepende yung type and function of a biosafety cabinet. Again, the blower and the filter. And in your class 2 cabinets po, um, the different types of class 2 cabinets po, ang kanila pong basis on differentiation is the blower and the filter. Specifically po, the placing of the blower and how the air is exhausted po and recirculated. Now in class 2A cabinets po, please take note that class 2A, when you hear the type A, so please take note that type A po, the exhausted air po is usually po exhausted outside the room. Okay? Pag sinabing air, air room. Okay? And then for class 2B cabinets naman, usually the exhausted air po is completely exhausted outside the building. Okay, it is hard ducted type of class 2 cabinet. Okay, again, class 2A, the air is exhausted in the room, while the class 2B naman po is exhausted, the air is exhausted using a hard duct. Okay, so when we say class 2A1 naman, when you hear A1, so usually the type 1 class 2 cabinets po, their blower po is usually located away from the filter. As you can see here, no, the class 2A1 and um class 2B1 have both um have both one in their typing because their blower po is away from the filter. Now when we say class 2A2 naman, so when we say 2, the blower naman is usually po near the filters. Okay? As you can see here, class 2A2 and class 2B2 po have the same placement of their blower which is near the filter. Okay? So kaya nga lang nagkaiba lang po yung class 2A1 and 2B1 because the class 2A1 has a closed exhaust system. Okay? Closed yung kanyang system. While the class 2B1 po have both closed and open exhaustion. Okay? So again, class 2A. Now usually po the air is exhausted in the room. A. And then yung 1 and 2 naman, when we say 1, the blower is away from the filter. The 2 naman is near the filter. Now, let's talk about po the filtering system naman po of your biosafety cabinet. So that part of the biosafety cabinet, the filters po, yun po yung main part ng biosafety cabinet natin that do the um, protection. Okay? Kasi po the filters po can provide decontamination of the air po supplied and recirculated inside the biosafety cabinet. And there are two types of filters po, standard filters po used in biosafety cabinets. These are your HEPA filters, your high efficiency particulate air filter, and another is your ULPA or ULPA or your ultra low penetration air filter. Okay? So ano lang po yung pinagkaiba po nila? So ang pinagkaiba po lang nila po, the two filters po have different filtering capacity when it comes to the different particles po in terms of size po which it can filter. Okay? So usually po kasi the smallest organisms po that it can filter po is your viruses. Now, in terms of filtering capacity po in terms of viruses, the HEPA po can only cover about half of the size of your viruses. So, hindi niya po nako-cover yung pinakamaliliit na viruses. But in your ultra low penetration air filters po, it can cover the entire size band of viruses. Thus, your ultra filter po gives an edge po in protecting the user and environment. But then again, your HEPA filter po is adequate na in most of the biosafety cabinets. So, in special work practices lang po ginagamit yung ating ULPA. Okay? So the most internationally recognized standard po for clean air po is your ISO 14644. So ito po yung fina-follow natin na international standard po when it comes to choosing what filter should be used in a biosafety cabinet or in a work practice. Okay? And under this ISO 14644, the ISO class of 5 po, yung ginagamit natin na standard for laminar flow cabinets, biological safety cabinets po, and pharmaceutical isolators. And under this ISO class of 5, no, we can determine po the efficiency of the HEPA and ULPA filters. Now, under the ISO class 5 and the study of US and European standard, the HEPA filter po has a 99.99% um, efficiency under 0.3 micron particles. Okay? And then in the ultra low penetration filter naman, it has 99.999% efficiency under a much smaller micron of particulates. It is under zero point 0.1 and zero point 0.2 to zero point 0.2 microns. So magsabihin lang po, it can filter up to that smallest size of particles. So that's why it is called an ultra low penetration air filter. Okay? And then last po for your filters, so here po shows you the summary po on um how efficient the two different filters po, okay, the um uh in terms of the different standardization. So in HEPA class, no, usually po ISO 524 po is applicable in HEPA filters, the type of work zone. So while your ISO 3 naman, yun po yung kino-cover ng ating ULPA. And they have usually same uh lifespan. Okay, they usually tend to go for 8 to 10 years, no, and they have also the same cost, no, when you want to replace these filters. Okay, and then nagkaiba lang po sila usually sa kaya nilang i-filter. So in terms of a million spores, usually in HEPA filter, 10 of the million spores can escape, while the ULPA naman only one, no, under a million spores can escape. That's how ULPA is highly efficient, okay, in filtering smaller substances. Okay. So besides knowing po the different safety protocols po and containment principles in controlling your microorganisms, one also method of controlling your microorganisms po is the different principles of killing the microbes po, which usually po using a sterilization and disinfection techniques. So on the succeeding slides po, we will now see the different principles, no, between each method of killing your microbes in terms of sterilization and disinfection. So, three terms po are commonly encountered when the topic po is about control of microorganisms, and that is sterilization, disinfection, and antisepsis. Now, in defining po sterilization, so sterilization po is the complete removal, no, or destruction of all microbial life, including bacterial endospores. Now, disinfection naman po, on the other hand, destroys only a defined scope po of microorganisms, including po some spores, but not necessarily all microbial life. Now, there exists the physical, no, and chemical methods of disinfection. Now, however po, most disinfectants are commonly, um, most of the disinfectants natin ay chemical agents po, no, applied to inanimate objects such as tabletops, laboratory hoods, and sinks. Antisepsis naman po is the term used when an agent is applied to the skin or living tissue po for the purpose of limiting or eliminating the number of of the bacteria present. Now, chemical agents used for antisepsis are called antiseptics. And it is important to note that antiseptics po are not sporicidal. Okay? So most disinfectants also are chemical agents which is applied po to inanimate objects. A substance po applied to the skin naman for the purpose of eliminating or reducing the number of bacteria present po is referred to as antiseptics. And take note, antiseptics again do not kill spores and cannot be used as disinfectants. In terms of a history of sterilization, disinfection po, and antisepsis, Ignaz Philipp Semmelweis po demonstrated the routine hand washing po that prevents the spread of the disease. And Joseph Lister naman developed the antiseptic system of surgery. He also introduced hand washing to British surgery, and he also discovered the phenol, okay, as an antimicrobial agent for surgical wounds and dressings. Okay, let's now see the important terms when it comes to antimicrobial agents. So when we say biocide class, it is a general term po describing a chemical agent po, and usually it is a broad spectrum chemical agent, no, that inactivates microorganisms po in a wide spectrum. So, mapa gram positive or gram negative, it can kill. Okay, when we say broad spectrum. So, biocides po can be as antiseptics, disinfectants, and preservatives. Okay, so when we say bacteriostatic naman, this is a specific term po used po, no, to refer the property of a biocide that can inhibit bacterial multiplication. Okay, so multiplication resumes upon removal of the agent when we say bacteriostatic. Bactericidal agents naman are um, uh, is a term, okay, used for biocides that can kill bacteria. Okay, bactericidal. Sterilization naman and disinfectants is defined na po kanina. Now, the term septic naman po is used to characterize the presence of pathogenic microbes in living tissues. And when we say aseptic naman, it is a term used naman to characterize the absence of pathogenic microbes. Now, the way no of eliminating this pathogenic microbes in a living tissue is called antiseptic. Okay, so antiseptic naman refers to the biocide or product no, that destroys or inhibits the growth of microorganism in or on living tissue when we say antiseptics. And preservation naman, it is a term naman used po or a process po used to prevent no multiplication of microorganisms in formulated products, no, including pharmaceuticals and foods. And antibiotics naman on the other hand are substances no that can be a substance that is naturally occurring or synthetic organic compounds which inhibits or destroy bacteria selectively. Okay? And that substance must be in low concentrations lang po to be safe on the surrounding tissue or in other normal flora, okay, na mga bacteria. Now, on this slide naman po shows you the different factors po that influence the degree of killing. Okay? So, first factor is the type of organism. Second is the number of organisms present. Third is the concentration of disinfecting agent. Fourth is the presence of organic material. Fifth is the nature of surface to be disinfected. Another is contact time, the temperature of the agent or the surface, the pH of the area or the disinfectant used, the presence of other factors such as biofilm formation, and the compatibility of disinfectants and sterilants. Now, chemical agents po are used no, mainly for disinfection and physical methods naman po are used mainly for sterilization purposes. However, no, some chemical agents po may also be used as sterilant. Now, the mode of action of some chemical agents may be one of the following. Now, first is reaction with the bacterial plasma membrane. Now, second is denaturation po or coagulation of bacterial proteins. And thirdly po is reaction with enzymes. And fourth is damage to nucleic acids. Now, damaging the cell membrane class may cause the contents of the cytoplasm po to leak out of the cell. That's why yun po yung may mga agents po tayo that target sa cell membranes. Ang pinaka main way po nila on killing the bacteria is destroying the cell membrane and then allowing the contents to be spilled out and then killing the bacteria. Okay? Now, the denaturation of proteins naman and the activation of enzymes will halt no, the bacterial metabolism. And then finally po, damage directed to nucleic acids naman will prevent bacterial replication. Now, as I have summarized po before, there are different efficacy no of these agents no that will depend on different factors no such as the concentration of the agents, okay, and the time of exposure. So there is a relationship po between the concentration of the agent and the time required po to kill a given fraction of a microbial population. Okay, time of exposure naman po, microbes are killed po kasi with a reasonable length no of time with chemical agents, and contact time is an important factor po talaga. Okay. So, usually kasi 'di ba may mga chemicals po tayo that targets the enzymes and then the metabolism. So, thereby kailangan po ng proper contact time and concentration. Temperature also is one of the factors that contributes to the efficacy of the agents to kill the microbes. No? Now, chemical agents po kasi are usually used at room temperature, okay, of 20 to 22 degrees Celsius. Now, generally, an increase in temperature leads to an increase in the activity of disinfectants, while decrease in temperature naman po leads to decreased activity. Now, the type and nature of the microorganisms naman po must also be considered. No? Different species may have different structures. For example, may mga bacteria po tayo that can produce spores. No? So, disinfectants may have varying results with each one of them. Now, more importantly po, organic materials po such as your blood, or pus po may inactivate some disinfectants. Now, this is therefore important po to lessen the amount of organic materials po from surfaces before placing a disinfectant to ensure effectivity. Okay. Now, added to these other factors is your pH. No. Now, the pH of material to be disinfected or sterilized can affect the activity po of the disinfecting or sterilizing agent. Now, it is critical to make sure at what pH po the agent is active and what the pH of the material to be exposed to the agent is at the time of the process will be done. Now, biofilms naman po, ano po ba yung mga tinatawag nating biofilms? Now, biofilms po, these are communities that are generally po layers of microorganisms that often po have a protective material over them, no, that shields the bacteria to be disinfected from outside environmental factors. And presence of biofilms po can increase, no, or decrease the efficacy of the agents to be employed with. Now, compatibility naman po of disinfectants and sterilants po. Now, some disinfectants naman po may inactivate, no, other disinfectants. For example, now the use of bleach and quaternary ammonium compounds together, no, may negate the activity of both disinfectants. Okay? Now, this diagram naman po arranges different organisms from most to least resistant. Now, we can see here po that prions in the topmost part, no. Prions po kasi are infectious proteins, no, causing degenerative disease of the nervous system, and they are resistant to heat, chemicals, and radiation. Now, on the right side naman po, we can see that the greater the amount of time given to an agent to exert its effect, no, the greater the number of microbes are killed. No? As you increase the exposure time, you also increase the number of microbes which are killed, no, in the process. Okay, so let's now see naman the applications of sterilization and disinfection. So why do we need, no, to practice proper sterilization and disinfection techniques po in the microbiology laboratory? Okay, so very critical po kasi, no, to maintain the sterilization and disinfection in the day-to-day task of a microbiology laboratory. Now, in the collection of our specimen, no, especially blood, no, for microbial analysis, aseptic techniques, no, must be strictly observed, no, before extracting blood. So you need to eliminate first the normal bacteria po on the skin of the patient, no, before drawing the blood, no. This is to lessen po the contamination of these insignificant microorganism. Now, instruments naman po in culture media must also be sterilized, no, because they may contaminate or our samples, no, leading to false result. Sterilization naman po and disinfection are also employed, no, for the safety nga po of the laboratory personnel and the patients. Now, disinfection po and sterilization po is divided into two groups. Okay, so sterilants or disinfectants can come with chemical methods and physical methods, and we will discuss po some of them. Okay. Now, in this table po shows us the different methods of sterilization disinfection, the physical and chemical agents that we will discuss po one by one. So, for physical agents po, summarized here po are your sunlight. Okay, sunlight is a type of a physical agent. Drying also, heat, which is divided into two subtypes, the dry heat and moist heat. Filtration also is a type of physical sterilization and disinfection technique. Radiation also, and ultrasonic and sonic vibrations. Under chemical agents naman, we will regroup the different chemical agents into which, no, into which po these chemical agents affect the bacteria and then thus killing them. Okay? So meron po tayong mga chemical agents that damage the cell membranes. These are your surface active detergents or disinfectants, phenolic compounds, and alcohols. Acids and alkalis naman po and alcohols can also damage po your proteins. Okay? And then, heavy metals, oxidizing agents, dyes, and alkylating agents naman po are agents that modify functional groups of proteins and also affecting the nucleic acids as well. Okay? So on the next slide po, we will now discuss first the chemical methods or chemical agents. Okay, so again, no, there are three classifications of chemical agents. Now, first, so those that causes leakage of cytoplasmic contents through interaction, no, with the cell membrane. Second, now those that interfere with bacterial metabolism by the coagulation of proteins, and denaturation of enzymes. And the third one naman po are those that modify the functional groups, no, of proteins and nucleic acids. So we know that when the nucleic acids are compromised, it may prevent bacterial multiplication. Okay, so the first one, no undersurface active agents or surfactants are cationic agents. So when we say cationic agents, they have positive ion in their structure. Now, surfactants po lower the surface tension of water, no enabling lipids po to be dissolved. Okay, so an example of this is the quaternary ammonium compounds or QUATs or QUATs. Now, these groups po of compounds causes disruption po of the plasma membrane, no, leading to leakage of important cellular contents. Now, however, quaternary ammonium compounds are not effective against tubercle bacilli and spore. Okay? And so they are mainly used for the disinfection of non-critical areas such as the floor, ano po ba, bench tops, and in restaurants. Now, another disadvantage po of this chemical group, your quats, no, is that they are inactivated by organic materials. Thus, now before you put quaternary ammonium compounds to disinfect the surface, you need first eliminate no the possible organic materials on the surface. For example, in the laboratory, so kung meron po itong mga blood or presence of serum and other organic materials, you need to first eliminate the organic material for it not to deactivate no the quaternary ammonium compounds. Now, Pseudomonas aeruginosa is known to be resistant to quaternary ammonium compounds. Now, under anionic agents naman po or those that have negatively charged groups in their structure are soaps and fatty acids. Now, these agents naman po are also known to disrupt the integrity of the cell membrane, again, no, leading to damage of the plasma membrane. Okay? So they are more active in gram-positive no, than in gram-negative bacteria, same those with cationic agents. And the difference lang po between cationic and anionic is that anionic agents po are active naman po at acidic pH, no? So, compared to cationic agents, they are more active naman po at a basic pH naman. Okay. Now, phenolics naman po are those with phenyl groups po in their structure and usually po are used mostly at hospital and household environments, and they can also be found in germicidal soaps. Now, such compounds po are active even in the presence of organic compounds. So, these phenolic compounds can be used po to disinfect surfaces with organic compounds. Now, phenol is a standard for the disinfectant screening test or it is called as phenol coefficient. So, hindi na po ito masyadong ginagamit as an antiseptic because it is a neurotoxic. Okay? So, ginagamit lang po ito to standardize, no? Or to measure bactericidal potency of new chemicals. This is called the phenol coefficient. Now, in this test lang po, the highest dilution of a disinfectant under scrutiny, no, that kills a bacteria after 10 minutes of exposure is determined. Now, in the same way, the highest dilution of phenol that kills an organism after 10 minutes is also determined. Then the potency po of the disinfectant is compared no, with that of phenol using a formula. Now, test organisms for phenol coefficient is S. aureus and S. typhi. So, ang ginagamit natin for phenol coefficient is Staphylococcus aureus for a gram-positive control. And the Salmonella typhi naman po is for gram-negative control na bacteria. In continuation po of the phenol coefficient, the calculated results naman po will then be evaluated. Now, if the value is greater than 1, no, the disinfectant is more effective than phenol. Okay? If the value naman po is equal to 1, then both of the disinfectant and the phenol have equal disinfecting powers. Now, if the value naman po is less than 1, now the disinfectant is less effective than phenol. Now, thus, the new disinfectant po cannot be used as a killing agent. Okay. Now, next naman po is your organic solvents. Example of this will be your alcohols. Now, your alcohols po are one of the most widely used disinfectant po in homes and in hospital settings. Now, alcohol po, no, disrupt lipid membranes and denature proteins as well. However, no, they are non-sporicidal but effective against Mycobacterium tuberculosis. And a disadvantage of alcohol po is that it is deactivated by the presence of organic material. Thus, you need to first eliminate the organic material before using alcohol. Now, ethanol is the usually used in the preparation of puncture site before blood collection. Now, it is active no against a wide range of bacteria, including gram-positive, gram-negative, and acid-fast bacilli. That is the Mycobacterium tuberculosis. And tandaan nyo ha, ulit, alcohol po is not sporicide. So, the uses of alcohol is that it is potent skin disinfectant. No, it is also a antiseptic as well kasi ginagamit ko siya for living tissues. So, disinfectants for clinical thermometers and other apparatuses. And please take note that the potent concentration of ethanol is 70% concentration. Okay, let's now proceed now to the chemical agents that denature proteins. And one example of this is your acids and alkalis. No? So acids and alkalis po also act as disinfectants, no? Because they denature protein and interrupting biochemical processes. However, no, the usefulness of some acids po as disinfectants po are limited due to their corrosiveness naman. Okay? So they cause conformational alteration of proteins, and then examples of acids are the following: your benzoic acid, citric acid, and acetic acid. Please take note also that acids po are helpful mo as food preservatives, which extends the storage life of food products. Okay. So third group naman po of chemical agents are those agents that modify the functional groups of proteins and also damages nucleic acid, now resulting to damage to bacterial multiplication. So one example of these are your heavy metals, no, your mercurials and your silver compounds. Now, heavy metals, no, tandaan nyo po, have been traditionally used as disinfectants. No, an example is your mercury. No, however, no, its use has been limited due to its toxic effects to the body. Examples of mercurials are the following: mercury chloride, and then organic mercurials. So they are used no usually as antiseptics. Examples of these are your merthiolate and mercurochrome. Okay, so another example of heavy metal naman po is your silver compounds. Example of this is your silver nitrate and silver sulfasalazine. Okay, so usually the uses of silver compounds are as ophthalmic and wound antiseptics for burn patients. So one example of a um a type of silver nitrate solution no used for as an ophthalmic solution po is your 1% silver nitrate. So this is also known as the Creed's prophylaxis, your 1% silver nitrate, and it has been used po as an eye drop solution, your 1% silver nitrate, against ophthalma neonatorum or ophthalmia neonatorum. So ophthalma neonatorum po is a conjunctivitis of the newborn. So nangyayari po ito when the birth canal of the mother po is infected with a bacteria called as Neisseria gonorrhea, which is a causative agent of Neisseria gonorrhoeae or the causative agent of gonorrhea. So, ang nangyayari po, yung baby po is infected with the bacteria in its eyes, no? And then, nagkakaroon pa ng ophthalmia neonatorum. Now, to treat this, you use 1% silver nitrate as Creed's prophylaxis. Okay? Okay, another chemical agents that disrupts nucleic acids and functional groups of proteins are your oxidizing agents naman po. So usually po, no, oxidizing agents po converts functional groups, these sulfhydryl groups po into non-functional disulfide groups. Now, one example of oxidizing agents are your halogens. Okay, now your halogens po are also useful chemical agents po for the control of microorganism. And two examples of halogens are your chlorine and iodine. Now, chlorine po is typically used in the form of hypochlorite, no? Such as sodium hypochlorite found in bleach. Now, the recommended contact time for bleach solution is about 3 minutes or longer in the presence of organic compounds that may inactivate them. Now, iodine naman po is also useful and may come in two forms. No? The two forms of iodine is your iodophores or iodine tinctures. Okay? Now, iodophores po is iodine combined with detergents, while iodine tinctures naman po are alcohol in iodine solutions. Now, iodophores are usually used po for disinfecting the sites of blood collection with a contact time of more than 30 seconds. So, bago ka kumuha ng blood sample for bacterial identification, you need to first disinfect the skin additionally with iodophores or iodine with alcohol. Now, hydrogen peroxide naman po is another type of oxidizing agent, no? And then it is used po for cleansing of wound, disinfecting medical surgical devices, and plastic contact lenses. Now, the potent percentage po of or concentration of hydrogen peroxide is in 3% concentration. So this is the active compound naman po or component of agua oxinada. Okay? And please take note also that hydrogen peroxide is active against vegetative and bacterial spores. So hanggang spore po kaya niyang patayin. Okay. So next naman po are dyes, no? Now certain dyes naman po or stains no, are also useful in the control of microorganisms. Beside po coloring, no, the surface of the bacteria, they can also kill the bacteria as well, no? So they are highly selective for gram-positive bacteria, and usually po they are used for the treatment of dermatologic lesions, and it is also used for formulation of selective culture media. Okay? So ginagamit din po siya, ina-add din po siya sa mga culture media natin to make it selective. Okay? Example of these dyes are your brilliant green, no, malachite green, and crystal violet. And they are all effective against gram-positive bacteria. Okay, next naman po is your alkylating agents. Now, example of unalkylating agents are your formaldehyde, butyraldehyde po, and ethylene oxide. Now, formaldehyde po class in liquid solution is commonly used as a tissue preservative. Although po high concentrations of formaldehyde can be used as a sterilizer, no, being an irritant po and a carcinogen limits its use. So, ang formaldehyde po delikado kasi it is an irritant and a carcinogen. Now, formaldehyde in gas form is used to sterilize biosafety cabinets. Now, the potent um, concentration po of formaldehyde is 37% in aqueous solution. Now, formaldehyde po can called formalin in this type of concentration. So tandaan niyo po ha, formaldehyde can also be used to prepare vaccines po from bacterial surfaces, viruses, and toxins. And formaldehyde is a bactericidal up until spores. Okay, next naman po are your glutaraldehyde. Now, the activity of glutaraldehyde po is it's sustained even in the presence po of organic matter. However po, it is only active in alkaline environment. So, mas mataas na pH. Okay? So, unlike other chemical disinfectants po that damages instruments, glutaraldehyde po does not corrode metal or rubber. So, it is the reason why it is the disinfectant po of use for some medical equipment. This is called sterilizer or is called sterilizing. Okay? So, ethylene oxide naman po is the most common gas used for sterilization. So, it is used naman po for materials no, that can be damaged by steam sterilization. So, petri dishes, catheters po are sterilized using ethylene oxide. Also, no, your disposable syringes are made up of plastic. So, that's why for it to be sterilized with, no, ginagamit po natin is your ethylene oxide. Okay, and the last one po would be your antiseptic agents. So pag sinabi lang pong antiseptic agents, again, these are chemical agents applied to the skin, okay, in animate objects. They must be able to able to control, no, or reduce the number of microorganisms in the tissue without harming the host cell, okay? So those that are used to prepare the site of blood collection are antiseptics. So ito po yung dapat na maging characteristic ng mga antiseptic agents. They can never be toxic to cell, they can never be corrosive, and they should never change the nature of the skin. Examples of antiseptics are the following: Savlon, no, which is chlorhexidine, alcohol in 70%, your iodine tincture, and your iodophor. Now, in this table po summarizes the different types of antiseptics and disinfectants together with their mode of action and application. Okay, let's just now move on to physical methods. Now, the first physical method of sterilization is your heat. Okay? Now, heat is the most commonly used po in controlling microorganisms. Now, heat usually comes in two forms. Okay? Either dry, no, or moist heat. Now, the basic difference between the two is that moist heat no, requires water or moisture, while dry heat naman po does not. Okay? And then the most reliable and universally applicable method of sterilization is heat. So dry heat po denatures proteins, while moist heat naman po denatures and coagulates protein because it uses water or moisture. Now, dry heat po can be used by the following methods. So first method of dry heating is flaming. Okay. Now, flaming involves a direct exposure of an instrument such as the bottles or tube necks, inoculating loops or wires to an open flame. So usually provided po by a Bunsen burner or alcohol lamp. A more safer no and recommended method po of flaming is using a Baptist incinerator. Okay, so direct flaming lessens the risk of contamination when microbes po are being handled for culture and biochemical testing. And please take note po, it is the efficacy of flaming po is uncertain. Okay, kaya hindi po siya ganun ka-effective in sterilizing. Okay, so ginagamit lang po siya for flash flash sterilization purposes, no, and then for aseptic techniques. Now, second naman po is using hot air oven or hot air sterilizer. So, hot air ovens po can also be used for sterilization of glasswares naman po, oil, no, petroleum, and or powders. Now, the temperature required is usually po 160 to 180 degrees with an exposure time po of 1.5 to 3 hours. Now, the first photo shows one application of direct flaming. Now, notice how the inoculating loop is exposed to the open flame. So, until it is red hot. Now, to ensure that the direct flaming sterilization is completely done. No? Usually sa mga metals natin, kailangan may red hot po. Or dapat red hot po yung ating wires. No? On the right, most photo naman po, we can see a back incinerator. Okay? So the hole in the middle of the apparatus is where the inoculating loops and wires are placed. Okay, let's now proceed now to moist heat. And then moist heat po kasi is the preferred um, type of sterilization when you are using heat no, due to more rapid healing. Okay? Now, moist heat po kills microorganisms through the naturation of proteins and enzymes no, and degradation of nucleic acids. And one of the most common application of moist heat is through boiling. Okay? Now, this is done by exposing the material to boiling water, so usually for 15 minutes. Now, however, it is not a reliable method of sterilization, no, because it does not kill bacterial endospore. So, boiling cannot kill bacteria that produces endospore. Okay, so this is used for sterilizing catheters, dressings, and fabrics. So, another method is tyndalization naman, a type of moist heat sterilization. So, it is also known as fractional, no, and or intermittent sterilization. So, this is done naman po using an instrument called Arnold's sterilizer. So this is called fractional sterilization po because this method po involves three separate heating cycles, okay, done within 3 consecutive days. Now each day class, now the materials to be sterilized are exposed at 100 degrees for 30 minutes. And then in this method po, the vegetative cells po are killed first in the initial process or cycle. Now the surviving spores would then germinate, not to become vegetative cells, which would be then killed po in the succeeding heating cycles na 100 degrees then for 30 minutes. So this usually is used for sterilizing heat-sensitive culture media na containing materials no, such as carbohydrates, eggs, or serum. Now, another method po of moist heat sterilization is your pasteurization. Now, pasteurization po is first developed by Louis Pasteur, no? Which is a heating process po that preserves the shelf life, no, of milk, wine, and other food products, no? It is used po to remove foodborne pathogens, no, and organisms responsible po for the spoilage of food. Now, there are three types of pasteurization class. So, the first is the low temperature holding or batch method, no? So this method destroys pathogens found in milk and reduces spoilage of food without affecting its taste. So the holding or batch method and the holding or flash method po, no, employs a certain type of temperature and contact time. Now, for the low temperature holding or batch method po, it is done po at 63 degrees for 30 minutes. Now, the second is the high temperature short time or flash pasteurization. Okay? So, ito naman po. So, the time of exposure naman po is and the temperature is 72 degrees now for 15 seconds. That is why it is called flash method. Okay? So, followed by immediate cooling. Now, the last one naman is the ultra-high temperature method. So in the exposure condition naman po at 140 degrees for 3 seconds. So again, the first is low temperature, second is the high temperature, and the third is the ultra high temperature, which employs 140 degrees for 3 seconds. Okay, and then again, the main uses of pasteurization are for milk, no, preparation of bacterial vaccines. Now, the next one naman po under moist heat is your autoclaving. And this is one of the most effective method of sterilization. And it is commonly used, no, in the microbiology laboratory. So ang principle nito is that it uses steam, no, under pressure. No, it is based on the principle that when the water is boiled, no, at increased pressure class, hot saturated steam will be formed, which penetrates and gives up its latent heat when it condenses on cooler objects, thus killing the bacteria inside out. Okay? So it destroys all organisms, no, vegetative or spores, no, syempre except prions, no, which are infectious proteins. Now the principle again of autoclaving is steam under pressure. So putting steam under 1 atmosphere of pressure or 15 psi achieves a temperature of 121 degrees. Now this temperature class all microorganisms again except for prions and their endospores are destroyed, no, within approximately 15 minutes of exposure. So usually ang setup ng autoclaving natin is dapat po nasa under ano po siya 1 atmosphere or 15 pounds per square inch at 121 degrees for 15 minutes. So that's the effective setup for autoclaving. Continuation of autoclaving class. So autoclaving again is a chamber which is filled with hot steam under pressure. So typically nga po, the exposure conditions is 121 degrees, 15 pounds per square inch, or square inch pressure, and then at least 15 minutes. Then the autoclave can be used for a variety of materials, such as glassware, culture media, and heat-stable equipment. So it cannot be used for equipment po na nade-destroy through heat ha, because it employs a higher amounts of pressure and temperature. So it can also be used to decontaminate medical waste. No? So but the conditions po must be 132 degrees, 15 pounds per square inch for 30 to 60 minutes. So again, for medical waste, tinataasan lang po yung temperature. It is 132 degrees, 15 pounds per square inch, and 30 to 60 minutes ang contact time. Now, the latent heat of evaporation is the amount of heat now that is required to convert boiling water to steam. Now, this conversion heat is quite large compared to the amount of heat that is required to make water hot. So that's why po mas effective po siya to kill bacteria. Okay? So again, it is used to sterilize solid and fluid culture media, medical and surgical equipment. So as we have discussed kanina po, no, your autoclaving is the most effective way. Kaya nga lang po, no, to ensure the um, na sterility, no, of the autoclaving process po, there are different methods po being employed during autoclave cycles po to ensure that the conditions po required for sterilization are met. For example, no, materials being sterilized po are taped with a piece of autoclave tape. No? When we say autoclave tape po, ito yung ginagamit natin to ensure that the proper temperature po is achieved. Okay? And the proper sterilization is achieved. And then ginagamit po ito together with the materials to be autoclave po in the chamber. Na at room temperature, the autoclave tape is white in color. However, when exposed po to the temperature of 121 degrees, yung black lines po will appear, no, throughout the length of the tape. Now, the appearance of the black lines then indicate that during the autoclave cycle, the ideal temperature was reached. Now, we can also use biological indicators, to assess the autoclaving cycle that has been successful. So they are called biological indicators because they are actual microbes. Now, usually those that produces spores. No? For autoclave class, the biological indicator is Bacillus stearothermophilus. Okay, again, the biological indicator for autoclave is Bacillus stearothermophilus. Now, the spores of the particular bacteria placed in either strip or liquid suspension in a vial are loaded together with the material being sterilized. Now, after the cycle of sterilization, the indicator is then incubated and observed for growth. Now, if growth is present, the sterilization cycle is successful. Okay. The last two type of moist heat sterilization or bactericidal bacteriostatic techniques are your freezing and lyophilization. Okay? Now, freezing po or cold temperature sterilization po is an important method po in preservation of food. So, it is bacteriostatic as it only reduces the rate of bacterial metabolism. However po, there are instances in which bacteria can be killed by cold temperature. Okay? So, take for example your bacteria Treponema pallidum. Now, your Treponema pallidum is a causative agent of syphilis. So it can be killed, class, when exposed to the temperature of 2 to 8 degrees for 72 hours. But the main principle of freezing is for preservation, for decreasing the metabolic activity of bacteria. So thus eliminating the rapid multiplication of bacteria. Now, next naman is your lyophilization or freeze drying. So, this is a process of freezing and drying po of materials as a means of microbial control. Now, it preserves din po bacterial culture for long-term storage. It involves rapid freezing and subsequent drying. Okay, in this diagram po shows you the different methods of heat application and the required conditions for maximum effectiveness. Okay. So another type of physical method besides dry heat and moist heat is your filtration process. So it is the mechanical sieving po through membrane filters. So typically, filtration po is used for sterilization po of heat labile and heat sensitive solutions such as vaccines, carbohydrates, antibiotic solutions, and others. Now, this is done by using membrane filters with pores. Okay, may mga butas po ito. So, thin membrane filters composed of plastic polymers po ito or cellulose esters containing pores of a certain size. Now, most bacteria, tandaan nyo, yeast and most po are retained by pore size of 0.45 and 0.80 micrometers. Tandaan nyo po ulit ha, they are retained. Ibig sabihin, they are filtered no through pore size of 0.45 and 0.80 micrometers. However, this pore size po may allow passage of Pseudomonas-like organisms because these Pseudomonas-like organisms have smaller size po. So usually po, for Pseudomonas-like organisms to be filtered, a smaller size of pores, usually po 0.22 micrometer size of pores, is available for critical sterilization. And membranes with pore size of 0.01 micrometer naman po are capable of retaining small viruses. So it can be also be noted that filtration can also be used to remove microbes from the air. Iba naman po ang ginagamit natin na type ng filters po dyan. So ang pinaka ginagamit po na filter is called HEPA filter or high efficiency particulate air filters for sterilization of air. Okay? This is usually used for biosafety cabinets. Okay? Now, HEPA filters are able to remove microorganisms larger than 0.3 micrometers and are used in laboratory hoods and in rooms of immunocompromised patient. So tandaan niyo po your HEPA po has 99.97 to 99.99% at 0.3 micron of efficacy. While your another type of filter is which is called your ultra low penetration air filters has higher filtration efficacy. So it has 99.999% at zero point 0.1 to zero point 0.2 micron efficacy. Okay? Now, another type of physical method is using radiation. And radiation po can also be used for sterilization. And there are two types of radiation. So, meron po tayong ionizing and non-ionizing radiation. Now, ionizing radiation po is typically short wavelength gamma rays, while non-ionizing radiation naman po is typically ultraviolet rays. Okay? And radiation destroys the DNA of bacteria, hampering their ability po to multiply. Okay? So continuing po your radiation. So please take note po that spore-forming bacteria are more resistant to ionic and ultraviolet radiation than vegetative bacteria because the spore coat confers protection and the DNA is different state in spore. So that's why radiation is non-sporicidal. So the usage of radiation are in the following: sterilization po of plastic syringes, catheters, gloves, and antibiotics. Okay? So non-ionizing radiations typically used in exposed surfaces such as operating room and laboratory rooms and can also be used for biosafety cabinets, your UV ray. Now, in this slide, we can see the recommended concentrations of different disinfectants. So these concentrations must be followed in order to ensure effective disinfection. So for example, your betadine or iodophor, the effective concentration of this chemical disinfectant is 2%. So while bleaching powder or calcium hypochlorite is in 14 grams in 1 liter of water. Now, the Dettol naman or chlorosilinol is in 4%. Ethyl alcohol is 70%. Glutaraldehyde is in 2%. Lysol is in 2.5%. Here's Savlon, which contains chlorhexidine and cetrimide. So, ang effective concentration naman po would be 2% to 5%. And sodium hypochlorite naman or your bleach solution is in 1% or 0.1%. So, in small amounts po or in disinfection of surfaces naman po, the effective concentration of sodium hypochlorite is in 10% din po. Now, in this slide naman po, we can see the recommended sterilization or disinfection methods po for different materials. So, inoculating loops and wires are sterilized using direct flame. Okay, we can see here. Glassware can be sterilized naman using hot air oven or with the autoclave. Now, heat-stable culture media naman po are usually sterilized using autoclaving. So, when we encounter naman po heat-labile materials no, or heat-labile culture media such as antibiotic solutions, serum, toxins, and your heat label culture media, usually they are sterilized po with the use of filters. Now, disinfection naman po of the skin before blood collection is always practiced. No, alcohols and iodine solutions po or both are typically used for disinfection of the skin prior to blood collection. Now, in this last slide naman po shows us the categorization po or classification of devices and the methods of effective disinfection of different devices. Now, hospital devices kasi are categorized into either a critical device, semi-critical device, or non-critical device. Okay? Now, critical devices po are those that enter sterile tissue and the vascular system. Okay? So, for example, like surgical equipment, needles, etc. And therefore, such critical devices po um, must be or do not contain no in every way possible all microbial life no must be eliminated from such devices. Semi-critical devices naman po are those that come in contact with non-intact skin such as wound or mucous membrane but do not penetrate them. So the recommended method naman for these devices are high level disinfection but still sterilization is preferred. Okay? So this diagram shows us the effectiveness of different methods of microbial control against spores, mycobacteria, naked or non-lipid viruses, fungi, and the typical bacteria. Now, for critical devices, it is important that all types of microbial life is eliminated as these may pose problems to the patient. So, for examples of disinfection methods for critical devices are the following: steam, no, dry heat, gas, chemical, and ionizing radiation. So all of these disinfection methods po for critical devices can kill up until spores. So they are called sporicidal. While your semi-critical naman po, the high-level disinfection for semi-critical devices are the following: using 2% glutaraldehyde, chlorine dioxide, and wet pasteurization are all types of high-level disinfection. And um, they are typically sporicidal except for wet pasteurization. For low-level disinfections naman po, tandaan nyo, these are disinfection methods po that are that are po non-sporicidal. So usually po they cannot kill up until spores. And examples of these are the following: sodium hypochlorite, quaternary ammonium compounds. Your alcohols po are not sporicidal. The phenolics po and iodophors are not sporicidal. That's why po low-level disinfection lang po ang application po ng mga disinfectants po na ito. And that summarizes or that's all no for our discussion for control of microorganism. And thank you everyone for listening.